Hi, good morning all of you students. This is Vishesh Rao, Botany faculty, Gosal campus, Sri Chaitanya Vijayawada. Today our botany topic is Microsporogenesis and development of male gametophyte. First of all, you have to learn the parts of flower in a flowering plants. Flower consisting of mainly two types of reproductive organs, such are accessory organs, another name non-essential organs, second category essential organs. These are the essential organs specially helpful for reproduction by producing gametes. And these are also called as the sporophylls or reproductive whorls. The floral leaves are found at the first two whorls are called accessory organs or non-essential organs or perianth members. Example for the perianth members, calyx and corolla. What is the calyx? Whorl of sepals is the calyx. What is corolla? Whorl of petals. The first whorl of the flower is calyx. Second whorl of the flower is corolla in a typical flower in flowering plants. These are generally sterile pods not directly involved in reproduction. Okay. Coming to the essential organs or reproductive organs. Third whorl of the flower is andesium. What is andesium? The whorl of stamens is andesium. Last whorl and innermost whorl of the flower is gynoecium. Whorl of carpels is gynoecium. Generally, these two are the male and female reproductive organs respectively. Okay. The andesium is the site for microsporogenesis and development of male gametophyte, and gynoecium is the site for megasporogenesis and development of female gametophyte. In this flower, sepals, petals, non-essential, stamens and carpels, essential organs. Andesium consisting of stamens. The number of stamens in a flower different from species to species. Another name of the stamen is the microsporophyll. It is the male sex organ or male reproductive organ. These are the stamens. In each stamen, two pods are there. The fertile pod or pollen producing pod is the anther. Second one is the staminal filaments. These two are the two important pods of a stamen. One is the filament and another one is the anther. Anther is a group of sporangias, microsporogias. Okay. Anthers generally two types. Majority of the dicots, majority of the flowering plants, anther is the dithecous. When you very few members like malvaceous members, example hibiscus plant, anther is monothecous. Anther is monothecous. Here, read the question. Dithecous anther is option 1 monosporangiate, second option bisporangiate, third option tetrasporangiate, last one divide of the sporangia. What is the correct answer? Tetrasporangiate. What is the meaning for the tetrasporangiate anther? In dithecous anther, transverse section at the center, sterile tissue is there. It is called connective. Other sides of this connective, pair of anther lobes are there. This is the one anther lobe. It is the second anther lobe. Each lobe possesses two pollen sacs, nothing but two thecas. The name of this chamber is the pollen chamber or pollen sac. Another name of the pollen sac or pollen chamber is theca. So total how many thecas are there? 1 and 2. Pair of thecas are found in each lobe of the anther. Every lobe of the anther in dithecous condition possesses two thecas. Due to the presence of two thecas in each lobe, this anther is named as dithecous. Okay. In second lobe also, another two thecas are there. Another name of the theca is microsporangia. Total how many microsporangias are there in each lobe? Two microsporangia. In overall dithecous anther, number of sporangias are 4. Hence, the name of this kind of dithecous anther is tetrasporangiate. Tetra, 4 sporangias. Next question. This is the one anther lobe. Second anther lobe. Number of microsporangias in each anther lobe 2. Second lobe also 2 sporangias. Total number of microsporangias in each anther lobe of the dithecous 4. It is the meaning for tetrasporangiate anther or bilobed anther. When anther is in young stage or earlier stages, consisting of homogeneous mass of the merisma tissue. It is a division gable tissue there inside of the anther. The name of the tissue is the sporogenous tissue. The sporon tissue encircled by multi-layered anther wall. Multi-layered anther wall. At the time of the maturity of anther, cells of the sporon tissue metamorphoses into form microspore mother cells. Another name of the microspore mother cell is PMC, means pollen mother cell. Both cells of the sporogenous tissue and spore mother cells are in diploid condition. These are able to perform meiotic cell division. Okay. It is the transfer section of the means a TS of dithecous anther. One microsporangia, second microsporangia, third one, fourth one. Inside from here to here, completely it is a multi-layered anther wall that is found around the sporogenous tissue. That is modified into megaspore mother cells. It is the TS of young anther for the 4 microsporangias. Next, how many types of layers are found in anther wall? Maybe either monothecous or dithecous. Types of layers in anther wall. Generally, 4 types of layers are there. What is the name of these 4 types of layers of anther wall? For example, this tissue is the sporogenous. The sporogenous tissue encircled by 4 layers of anther wall, 4 types of layers of anther wall. That innermost layer is tapetum. Innermost layer of the anther wall is the tapetum. That is the binucleated. The layer next just outside to the tapetum layer is the middle layers. Middle layer, one more middle layer, one more middle layers. Next layer and thickest layer of the anther wall is the endothecium. One cell, second cell, third one, fourth one, fifth one. The cell walls of this endothecial layer are heavily thickened due to the presence of cellulosic fibrils. Name of this layer is endothecium. Outermost layer of anther wall is the epidermis. One cell thickened layer. All the cells are present in only one row or one cell thickness. These are 4 types of layers in anther wall. Outermost layer is epidermis. Second layer endothecium. Third one middle layers. In this middle layer anther wall, number of layers are more than 1. Innermost layer of the anther wall is the tapetum. Okay. Now 4 layers together called anther wall. Each microsporangium possesses mainly 2 structures. A diploid tissue found inside of the microsporangium at young stages is the sporogenous tissue. The sporogenous tissue encircled by 4 types of layer contained anther wall. Okay. Such anther wall layers are epidermis, endothecium, middle layers, tapetum from outside to inside are centripetal arrangement of these 4 layers. There is another important question here. Among these 4 layers, first 3 layers are essential for dehiscence of anther. Okay. Tell me once again, what layers of the anther wall helpful for the dehiscence of anther to liberate pollen grains? Outer 3 layers. Functioning of such layers, dehiscence of anther wall. Okay. Name the layer of, one more question, name the layer of anther wall that is helpful for providing nourishment for the development of pollen grains from sporogenous tissue, tapetum. It is highly nutritive. Number of nutrients are many here. And each cell possesses either 2 or more than 2 nuclei are there. It is about the tapetum. This tapetum layer is absent when anther reaches to maturity by providing food material. Due to supplying of food material for the development of microspores from the sporogenous tissue, this entire tapetum layer gradually perished out or depleted. It is about the functioning of anther layers. Observe here in second figure, pollen liberation occurs due to the maturity and anther already dehisces here. What is the site of dehiscence of anther? In each lobe, it is the one lobe, each lobe 2 thecas are there, or 2 microsporangias. Between the 2 microsporangias, one longitudinal groove is there. That longitudinal groove is the line of dehiscence. Line of dehiscence. In second lobe also, between the 2 microsporangias, one more longitudinal groove is there. It is also a line of dehiscence for the breakage of anther. Then all the pollen grains developed in these 2 microsporangias of a lobe exit out through the common way. It is the common exit for the liberation of pollen developed from these 2 microsporangias of a lobe. So same kind of story is there in second lobe also. Okay. Total how many exits are there in this dithecous anther? 2 exits. I want to read this question. The part of the pollen grain which is thick and rigid with various ornamentations is chiefly made up of sporopollenin. In structure of the pollen grain, only 2 wall layers are there. Innermost wall layer, thinnest layer, that is the intine. Outermost thickest layer is the exine. Chemically, this exine is made up of sporopollenin. The question is clearly mentioned. It is the thickest layer, hard and rigid layer due to the excessive deposition of the sporopollen material. And different types of orientations, ornamentations are there in the outer, thickest, rigid exine layer. Anther is broad, knob-like fertile pot. Typical angiospheric anther has the 2 lobes. 2 lobes are very common. Such anther is called bilobed, and due to the presence of 2 microsporangias or thecas in a lobe, it is also called as dithecous. Already you learned about the tapetum. Tapetum is the innermost layer, and functioning of the tapetum is the providing nourishment, and number of nucleus in the tapetum are more than 2. That is the important character or peculiar character of the tapetum. What is the reason for presence of more than 1 nucleus in tapetum layer? Due to the free nuclear division, means in the cell division mitosis, only karyokinesis occurs without cytokinesis. That is the reason for formation of more than 2 nucleus in a tapetum. One more question. In flowering plants, mature male gametophyte is derived from a pollen mother cell. By means how many mitotic and meiotics are required to form a mature male gametophyte from pollen mother cell? Listen. It is a one pollen mother cell. Another name of the pollen mother cell is the microspore mother cell. This microspore mother cell undergo 1 meiotic division, nothing but reductional division. Then it gives rise to 4 pollen grains. Pollen grain 1, 2, 3, 4, all are haploid. 4 pollen grains. Each pollen grain undergo 2 mitotic divisions to form mature male gametophyte. Mature male gametophyte. This is the structure of male gametophyte. Intine of a pollen grain. The intine involved in formation of pollen tube, exine, and maximum number of cells in this pollen tube 3. One is a vegetative cell, remaining 2 are sperms. The name of entire this structure is mature male gametophyte. The maximum number of cells in this mature male gametophyte are 3. The 2 are male gametes, another name of the male gamete sperms, another name of the vegetative cell. To form this mature male gametophyte, remaining required number of mitoids are 2. Meiotic division is the 1. Then what is the right answer? 1 meiosis, 2 mitosis, to yield only 1 mature male gametophyte from a pollen mother cell. Then the right answer is second option. 1 meiosis, 2 mitotic divisions. Yes. The next question. Each microsporangia of the dithecous stamen or anther consisting of 10 pollen mother cells. What would be the total pollen produced from that flower? If that flower consisting of 5 stamens. All the stamens of a flower androecium. A number given as a 5. 1 stamen with a dithecous anther. The dithecous anther. Microsporangia 1, 2, 3, 4. According to question, how many pollen mother cells are given in each microsporangia? 10. Second microsporangia, 10, 10, 10. In every one stamen, total number of microspore mother cells are 40 according to this question. Microspore mother cells are 40. Whereas in 5 stamens, 5 40s are 200 microspore mother cells. 200 microspore mother cells. But what is the main key point in this question? How many pollen obtained from this entire flower, including 5 stamens? Each microspore mother cell can yield 4 pollen grains. But total number of microspore mother cells in this 5 stamens, 200. Then 200 into 4 pollen grains. Total number is, means in entire that flower, total or maximum number of pollens are 800. Right answer is the third option, 800. Okay, clear? Listen once again. In this flower, given number of stamens 5. In each microspore mother cell, given number of microspore mother cells are 10 in each microsporangium. The number of microsporangias are 4 in each stamen. So, one stamen forges 10 into 4, 40 microspore mother cells. But entire flower, 200 microspore mother cells. The entire flower consisting of 5 stamens. In each stamen, the number of spore mother cells 40. 5 fours, 200 microspore mother cell. Every spore mother cell able to give rise to 4 pollen grains or 4 microspores. So 200 into 4, 800 pollen grains obtained from this kind of flower according to this formula, numericals. Okay, clear? Next question. What is the meaning for microsporogenesis? Production of or process of formation of microspores from microspore mother cells, okay, or pollen mother cells through the meiosis is called microsporogenesis. Simple, formation of microspores inside of the microsporangium or anther from the spore mother cells is another sporogenesis or microsporogenesis. Okay, what is the site of microsporogenesis? Microsporangia found in anther. Likewise, one more question. What is the site of megasporogenesis? Megasporangia. Megasporangia is nothing but ovules. Ovule is the site for megasporogenesis. Anther or pollen chamber is the site for microsporogenesis. Where the sporogenesis occurs, there automatically meiosis occurs. So, anther and pollen grains both are sites for meiotic cell divisions. Okay. Next slide. These are the 4 microspores obtained from one mother cell are in cluster form. Hence, at maturity, this microspore mother cell can yield 4 microspores. At the beginning, these are the 4 microspore or daughter cells obtained from one mother cells are in closed condition or fused condition. The name of the structure is pollen tetrad or microspore tetrad. Pollen tetrad or microspore tetrad both are same. Observe here, it is the pollen mother cell. At the end of the meiosis 1, it gives rise to dyad, 2-cellular structure. At the end of the meiosis 2, it gives rise to 4-celled structure. The name of the structure is tetrad. If the mother cell is the pollen mother cell, hence the name of this tetrad is pollen tetrad or microspore tetrad. Okay. Later, at maturity, these are the 4 microspores separated from tetrad condition. Then each and every microspore develops into pollen grain. This entire process is called microsporogenesis. The site of this process is microsporangium or pollen chamber or pollen sac found in anthers of a flower. Try to solve one more question. Starting with the microspore mother cells. What is the least number of, means the minimum number of meiotic and mitotic genes required respectively that could occur during the formation of 400 male gametes in angiosperms? What is the key point in this question? 400 male gametes. To yield 8 male gametes. 8 male gamete is the 1 unit. How many number of pollen grains needed? 4. Each pollen can yield 2 male gametes. 4 pollen, 4 twos are 8 male gametes. 4 pollen grain obtained from single mother cell. Only one pollen mother cell undergo reductional division or meiotic cell division to yield 4 pollen grains. 4 into 2, 8 male gametes. So, total number of male gametes obtained from one pollen mother cell, H. The ratio of pollen mother cells to the number of male gametes 1:8. 1:8. Already earlier you learned number of mitoid divisions in each pollen grain, 2. So, here the number of pollen grains are 2. In each pollen grain, number of mitoid divisions 2. 4 into 2, 8 mitotic divisions. Here one simple thing is there, observe. Number of male gametes always equal to the number of mitotic cell divisions. Okay. So, what is the ratio of number of meiotic and mitoid divisions occurs during the development of 8 male gametes from one pollen mother cell? 1:8. 1 meiosis, 8 mitotic cell division. How can you say here the 8 mitoid divisions occurs? In each pollen grain, 2 mitoid divisions are there. These are the 2 mitoid divisions in each pollen grain may not occur at a time. Observe here, I will explain detailedly about these 2 types of mitotic divisions in a pollen grain. It is the double layered pollen grain. In the single haploid nucleus is there with dense cytoplasm. Due to the first mitotic cell division, it is the first mitosis. We can get a 2-celled pollen grain. It is also pollen, but it has the 2 cells. One is the generative cell, another one is the vegetative cell. Vegetative cell is the bigger cell, generative cell is the smaller. And it seems to be as a floating cell inside of the cytoplasm of vegetative cell. We can make one more question here. Name the cell is found in another cells. That is the generative cell is found inside of another cell, vegetative cell. Okay. Up to this development, only one mitotic cell division completed. Next, the second mitotic division in the same pollen grain. Generative cell undergo one more mitotic cell division. That is the second mitosis. Give rise to 2 sperms. Another name of this sperm is male gamete. Finally, tell me how many mitotic cell divisions are needed for to yield 2 sperms from one pollen grain? 2. One is the first mitoid division in the haploid nucleus of the pollen grain. Second mitoid division occurs in generative cell of the 2-celled pollen grain. Total number of mitosis 2 in each pollen. That's why here it is clearly mentioned 4 into 2. 4 is the number of pollen grains, 2 is the number of mitoid divisions. Total number of mitosis 8 during the development of 8 male gametes from one pollen mother cell. Apply these concepts to solve this question here. To yield 400 male gametes, how many mitosis and meiosis required? 50 meiotic cell divisions, 400 mitotic cell divisions. Total required number of pollen mother cells 50. These 50 cells undergo 50 meiotic, nothing but reduction divisions. Then total number of pollen grains in each pollen mother cell 4 pollen grains. 54s are 200 pollen grains. How many male gametes obtained from each pollen? 2 male gametes. 4 into 2, 8. From the 200 pollen grains, 400 male gametes. In each pollen, 2 mitosis. Whereas in 200 pollen grains, number of mitotic divisions 200 into 2, total 400 mitosis. That's why answer for this question is 50, 400. 50 meiosis in 50 pollen mother cells, 400 mitosis in 200 pollen grains for the production of 400 sperms or male gametes. Okay, right answer is the 4th option. Coming to the next question. In the region of the germ pore, the exine of the pollen grain is. In the region of germ pore, the The exine of pollen grain is thin. Second option. It is the innermost layer of the pollen, intine. Outermost layer of the pollen is exine, it is the thickest layer. But the thickness of the exine layer may not uniform. At some areas, very thin areas are there. The name of that thin area is the germ pore. The germ pore facilitates pollen germination by forming pollen tube. Okay. Once again, give the definition for the germ pore. It is the thin area of the exine in pollen is the germ pore. Observe here. Diagrammatically, these are the germ pore, germ pore, germ pore. Except the germ pore, remaining entire area of the exine, thickest sporopollenin portion is there. Okay. In the germ pore only, thin area or less amount of sporopollen is deposited. That sporopollenin provide protection for the pollen grains from high temperature, strong acids or alkaline medium or another adverse conditions. It is the reason for survivability of pollen for the long period in external adverse conditions. The thing is mentioned here. Because of the presence of the sporopollenin, pollen grains of the fast plants are well preserved as a fossils. Study of pollen is the palynology. Study of pollen grains is known as the palynology. Observe here, 5 sequential diagrams are there. First one, single nucleated pollen, just recently formed pollen grain. Whereas in second diagram, two-celled condition is there. Means a partially developed male gametophyte. Partially developed male gametophyte. There is no much variation between second and third diagram. Okay. This kind of development of male gametophyte occurs before pollination. Generally, pollen liberate out from anther at two-cellular condition, majority of the angiosperms at two-cellular condition. The two cells are, one is the vegetative cell, or another name of the vegetative cell is the tube cell. Second one is the generative cell. Anther dehiscence at this stage and two-celled pollen liberate out and reach the stigma with the help of any kind of agent. After pollination, remaining development of male gametophyte occurs. After pollination, the first stage is formation of pollen tube from intine. Then both the cells of the pollen grain, generative cell and vegetative nucleus will enter into or migrate into this pollen tube. After entry of the generative cell into pollen tube, in that pollen tube place, the generative cell undergoes second mitotic cell division to yield two male gametes or sperms. This entire phenomena or development or process is called as development of male gametophyte. From this organization or mechanism, we can make several questions. Question number 1, simple question. What is the first cell for the development of male gametophyte? Pollen or microspore. Another name of the or earliest structure of the pollen grain is microspore. That microspore or pollen is the first cell for development of male gametophyte. Especially in angiosperms, how many mitotic divisions occur during the development of male gametophyte from a pollen? 2 mitotic divisions. First mitotic division in single haploid nucleus of the pollen. Second mitotic division occurs in generative cell, that is the place that are located in pollen tube. Okay. This entire male gametophyte development may divide into 2 parts based on their occurrence or place. One is the before pollination. Second phase of the development of male gametophyte is development of male gametophyte after pollination. Before pollination, 1 mitotic cell division. After pollination, second mitotic division. In most of the angiosperms. Whereas in some kind of another kind of angiosperms, less than 40% of angiosperms, these are 2 mitotic divisions takes place before pollination. There is no mitotic cell division after pollination. This is a slide can explain both the things at a time, microsporogenesis and development of male gametophyte. All these are the microspores or pollen grains. Development of microspores from anther or stamens of the flower is known as the microsporogenesis. From here to here, it is a completely microsporogenesis. The result of the microsporogenesis is formation of pollen. That pollen is the first cell for the development of male gametophyte. So, this entire diagrammatic presentation given in this slide can explain both microsporogenesis as well as development of male gametophyte. Okay. Now this is the structure is the fully matured male gametophyte or fully developed male gametophyte. Maximum number of cells in male gametophyte, one is the vegetative cell, remaining two are sperm cells, 1 plus 2, maximum number of cells 3. It clearly explains gametophyte is highly reduced in angiosperms when compared to the remaining plants in the plant kingdom. Generally, pollen are more beneficial for mankind, even though some kind of advantages are there. That one of the disadvantages of the pollen grain, it causes allergy. The allergy causing pollen grains commonly found in carrot grass. Botanical name of the carrot grass plant is Parthenium. Observe here, this is the Parthenium image. It is not indigenous. It is imported in India from Europe countries at the time of introduction of wheat grains from foreign countries. Okay. The pollen of this Parthenium plant, common name is the Congress grass, causes for long-term chronic bronchitis or asthma. Asthma disease causing pollen, commonly found in carrot grass. Scientific name of the carrot grass, Parthenium. Advantages of the pollen grains. Pollen grains are rich in nutrients, especially proteins and some other vitamins. They are taken as a tablets or syrups, other kind of food supplements for health conditions. All these are the pollen products. These pollen grains available from the market in the form of pills or capsules or tablets or powder or syrups also. All these are products of the pollen which are more beneficial for our humankind or human health. Pollen consumption has been claimed to enhance the performance of the athletes and race horses. To improve the efficiency of the race horses and athletes, pollen grains are used as a best nutritive supplements. That is one of the major advantages of pollen for human welfare. Yes. Read this question and try to solve the answer. In cereals like rice, wheat, maize, the viable period of the pollen grain is 30 minutes, half an hour only. Means after liberation of pollen from by the dehiscence anther, it should reach to stigma within 30 minutes. Otherwise, the germination capability will last. Okay. So the viable period of the pollen in cereals 30 minutes only. That viable period is different from species to species. Least viable period found in cereals. Highest viability found in Leguminosae members like pulses, Solanaceae members, and Rosaceae members. It is the image of the cereal, wheat, rice. In that pollen viable period, just half an hour only. Whereas, Rosaceae members, Leguminosae members, and Solanaceae members, the viable period, the few months. Next, it is possible to store pollen grains of large number of the species for years in answer liquid nitrogen, because the liquid nitrogen provide minus 190 degrees centigrade for the proper preservation of pollen without losing their viable period. Okay. At what temperature pollen grains are stored for a long time in laboratory? Minus 190 degrees centigrade. That much of temperature provided through the liquid nitrogen. This kind of preservation of the pollen in the liquid nitrogen is also called as cryopreservation. Also called as cryopreservation to maintain pollen banks. It is the cryopreservative labs. In this large tank or chamber, liquid nitrogen is stored for the preservation of pollen grains. Identify the wrong statements from the following. Read the statements one after another one. Intern of the pollen grain is made up of sporopollenin. Intern made up of pectocellulose, not sporopollen. That's why statement 1 is wrong. Pollen grains are well preserved as a fossils because of the presence of sporopollenin. Hardest organic material. True. Enzymes can degrade the organic material of the Exine of pollen grain. True. It is the false. Enzymes cannot degrade or digest the sporopollen material found in Exine of the pollen grain. Sporopollen can withstand at the high temperature and strong acids and alkaline substances. Statement true. Then the question is about incorrect statement. First statement and third both are wrong. That's the right answer is third option. Next question. Identify the correct match or correct statements. More than 60% of angiosperms pollen grains are shed at two-celled condition, not three-celled condition. That's why it is the false statement. And 40% of angiosperms pollen grains are shed at two-celled condition. No, it is the three-celled condition. In most of the angiosperms, pollen grains liberate out two-cellular condition. In few angiospheric plants, pollen liberate out at three-cellular stages. More than 40% of angiosperms pollen are shed at four-celled condition. Wrong. In pollen grain, max number of cells 3 only, not 4. So statement 1, 2, 3 all are wrong. Fourth one, nearly 40% of angiosperms pollen liberates at 3 cellular stage. So D right answer. Then what is the correct answer for this question? D only. Only one statement is correct. Next question, pick out the true statements. The spore mother cell in angiosperm is the microspore mother cell. True. Microspore mother cell is another name. Okay, sorry. Microspore mother cell is another name of spore mother cell, not sperm mother cell. Is one misleading term is given. Sperm, not spore. That's why first statement is wrong. Okay. I think you get a clear idea about it. It is not a spore. Sperm is given. That's why this statement is wrong. Megaspore mother cell is also called as mother cell of embryo sac. Wrong. Embryo sac is the female gametophyte. What is the first cell for the development of female gametophyte? Megaspore, not a megaspore mother cell. Embryo sac is in haploid condition. Mother cell should be in haploid condition. But whereas here, megaspore mother cell is in diploid. So it is not a megaspore mother cell. Only megaspore is the first cell for embryo sac. That's why it is also a wrong statement. Microspore is the mother cell for male gametophyte. Wrong. Microspore, haploid, male gametophyte. Haploid, that's why the statement is true. Microspore is the first cell for the development or mother cell for the development of male gametophyte. Clear? Microspore nothing but a pollen grain. After the complete development of gametophyte, it becomes like a germinated pollen grain. Another name of this germinated pollen grain or pollen grain with a tube with 3 cells is the mature male gametophyte. For this male gametophyte, mother cell is pollen. Another name of it is microspore. That's why true statement is third option. Zygote is the mother cell of sporophyte. Sporophyte is the plant body. The first zygote is the first cell for the development of sporophyte. So it is also true statement. So what is the right answer? 3 and 4. True. 1 and 2 false statements. Next question. Assertion reason type. Capital cells are binucleated. The number of nucleus are either 2 or more than 2. True. What is the reason for presence of more than one nuclei in each cell of the tapetum? Free nuclear divisions. Means the karyokinesis is not followed by the cytokinesis. For example, it is the cell. It has a one single nucleus. After KK means karyokinesis, it gives rise to two nuclear condition. There is no cytokinesis here. Without following the cytokinesis, again the two nucleus undergo further nuclear divisions. Such kind of divisions are called free nuclear divisions. Free nuclear divisions always leads to sinusoidic or multikaryotic condition. Then what is the correct answer here? Assertion and R correct. R is the correct explanation. A. Next, the pollen tube wall is made up of pectocellulose. Statement true. Because the tube is formed from intine of the pollen grain. Intine is chemically made up of pectocellulose. The structure developed from intine is also made up of pectocellulose. Also, right answer is first option. Reason explains assertion. Next question. Pollen are well preserved in fossils due to the presence of sporopollenin. That is the resistance to microbial activity or microbial decomposition. So here also the right answer is both A and R or 2. R is correct explanation of the assertion. Gametophytic generation starts with gamete. Second statement, sporophyte generation starts with zygote. Statement 1 is the false. Gamete is not a first cell for the gametophytes. For example, if the spore is there, then only the statement is correct. Spore is the first cell for the gametophytic generation. Okay, zygote is the first cell for the sporophytic generation. Statement 1 is false, second one is the true. Okay, thank you. I will start the megasporogenesis in next class. Bye.